Right. අපි ගිය සතියේ that we have discussed about the longitudinal view and the location of the nephrons in kidney. Can you remember it to us?
Yes.
Have you done the homeworks? I gave it you to draw the figures. කරාද?
කරන්න බැරි වුණා.
කරන්න බැරි වුණා. Right. ඔයා අනිවාර්යෙන්ම කරලා දාන්න ඕනේ. හෙට ඔයාට හෙට time තියනවද?
ඔව් සර්.
right. එතකොට හෙට වෙද්දි දාන්න ඕනේ මේ figures ටික. අනිවාර්යෙන්ම දාන්න ඕනේ මොකද ඔය කියන්නේ exam එකට එන තැනක් මේක. Okay දුව?
Okay සර්.
right. හරි. එතකොට දුව අපි අදට දැන් ඔයාට මතකද that the part of the nephrons, structure of the nephron?
ඔව් සර්.
right. අපි මේ parts ටික ආයෙ කියවමුද? මතක් වෙන්නත් එක්ක.
හරි සර්.
right. පටන් ගන්න.
Glomerulus, Bowman's capsule, renal artery, renal vein, descending limb, ascending limb, blood capillaries, loop of Henle, collecting duct, distal convoluted tubules.
right. එතකොට glomerulus, Bowman's capsule, renal artery. That renal artery supplies the blood to the nephron, and that blood will flow out through the renal vein, right? And the nitrogenous waste and the other excretory products that carries through the renal artery will be filtered in the glomerulus, and those products will be collected in the Bowman's capsule, and blood which are flowing through the renal vein will not have that excretory products. Clear?
Yes.
It will be filtered out. Right. Then now after that, after all, the collecting duct will collect the at the filtrate of the other nephrons, and it will go to this pelvis part. After that, it will go through the ureter and to the bladder. Clear?
Yes.
So urine will be passed out from the bladder at the end. Right. So now, right. That's the general process. But this general process is little bit complex with the process of urine formation. This process of urine formation is very that means much needed part for your audience, right? Right. Yes, that I will describe it first, then we can go to the questions, right? This process of urine formation is consist of 3 steps, right? First one is ultrafiltration. Second one is selective reabsorption, right? Third one is secretion, right? So this ultrafiltration is a filtering process, right? That means uh, that the blood carries to the renal artery, right? Should be filtered out, uh, and that filter is again undergo some reabsorption process, then to the secretion, and then to the to form the exact urine, right? Right. Now, when we are discussing the ultrafiltration, this afferent arteriole, right? This afferent arteriole supplies the blood to form these capillaries inside the uh, Bowman's capsule, right? That means this afferent arteriole is dividing into network of, that means bed කියලා කියන්නේ a bed of capillaries, right? In the uh, what in the Bowman's capsule. Then those capillaries will collect again to form this efferent arteriole. But the difference between this efferent arteriole uh, and the afferent arteriole is it's that the efferent arteriole diameter is smaller than the afferent arteriole. Afferent එකේ වැඩි. Therefore, it will build a pressure inside these capillaries. Why? This entering volume of blood cannot maintain the same speed in the efferent arteriole. මොකද පොඩි හරියට නිකන් ලොකු පාරකින් එන වාහන ටික ඔක්කොම එකපාරකට පොඩි පාරකට දැම්මා වගේ. අපි හිතමු හයිවේ එකෙන් ගොඩාක් වාහන එනවා. ඇවිල්ලා ඒ වාහන ඔක්කොම පොඩි පාරකට දානවා. දැන් අර එනවා නේද ට්‍රැෆික් එක. වාහන හිර වෙනවනේ පාරේ කැපිලරිස් වල නේද? අන්නේ ප්‍රෙෂර් එක තියෙන්න ඕනේ මේ ෆිල්ටරින් ප්‍රොසෙස් එකේ. හරි? දැන් මේ හරිය බ්ලඩ් හිර වෙනවා. හරි? clearද දුව?
Clear, sir.
From this figure also you can see this difference between the diameter. This afferent arteriole is larger than the efferent arteriole. Right? So what is this afferent arteriole? Afferent arteriole is a direct branch of the renal artery. Right? Renal artery is dividing to make the afferent arteriole. Yeah. After that, these efferent arterioles are collecting to make the renal vein. Right. It's about the afferent and the efferent arterioles. Then this filtering process is happening here. Right? Right. This right, that we minister is specific the glomerulus. Right? Glomerulus is the dense network of capillaries in the Bowman's capsule. Right? Then you know that blood is filtering out. That filtered blood is called what? Glomerular filtrate. Right? glomerular filtrate. This process is called what? Ultrafiltration. Right? So this filtered blood will be collected to the Bowman's capsule. Now the problem is it is not a problem actually. That's the nature. This large molecule, right? Like plasma proteins, you will learn about the plasma proteins in the future. And the blood cells will not be filtered to into the glomerular filtrate because that filtrate is produced by the capillaries. They are very thin. One cell layer thickness එකක් තියෙනවා. Right? So these large molecules will not be filtered. Just the small molecules will be filtered. Right? This glomerular filtrate is very similar to the blood plasma. Blood plasma. That you know that our blood can be divided into two parts, plasma and the blood cells. Right? This blood plasma contains much amount of water. Right? So this filtrate is also containing, right, water, right, mainly. And the glucose, right, is those small molecules are filtered out. Glucose, amino acids, vitamins, drugs, and other ions, right? All those things are filtered through the what this uh, glomerular glomerulus, right? Then, but when you are talking about this filter rate, right? You can see some essential molecules. What are they? In the glomerular filter rate. Oh, I am asking what are the essential molecules for our body, which are there in the glomerular filter rate.
Glucose, amino acid, vitamins.
Right. And water is also much needed, ne? And the hormones, right? What about the urea? What is it? Sure. Urea ආව නේද?
නැහැ.
Urea කියන්නේ මොකක්ද?
excretory product එක.
මොන excretory product එකක්ද?
kidney වල.
ඔව් මොනවද ඒකේ තියෙන්නේ? Nitrogenous waste එක නේද? Urea වල තියෙන්නේ. හරි? Nitrogenous excretory product එක. right? ඒ urea ගොඩක් එකතු වෙන එක අපේ ඇඟට හොඳයිද නැද්ද? Toxic. හරි? ඒ හින්දා ඒවා අයින් කරන්න ඕනේ. ඕකේද?
හරි සර්.
right. හරි? right. Then so there should be a reabsorption process නේද? That because
ඔව්.
many essential molecules are already filtered through this glomerulus, but it should be reabsorbed, right? Then should be reabsorbed. Now we are in the selective reabsorption process, right? Right. These molecules are reabsorbed to the blood capillaries. Thin blood capillaries are there among among the among the nephrons. So these blood capillaries are the ways for the that reabsorbed molecules, right? 90% of water is reabsorbed. All the glucose is reabsorbed. Amino acids will be reabsorbed because we want amino acid to produce what? We need amino acid to produce
Protein.
proteins, right? Very good. And the vitamins, right? Some amount of salts. And the small amount of urea is also reabsorbed. Urea, uric acid, that that yes, they are nitrogenous excretory products, but they are also reabsorbed, right, into the plant. Then the composition of this glomerular filtrate will be changed because all the things are reabsorbing. So this composition is changing, right, of the filtrate. I තියෙන දේවල් ගත්තාම composition එක වෙනස් වෙනවා. හරි. Then this glomerular filtrate is released into the collecting ducts. Then into the pelvis, like I said previously, right? Pelvis is here. This is the pelvis part, right? All the things will be connected to the pelvis with the collector part. Then to the ureters and to the bladder, right? Usually, that if you are looking for the volumes, right? That the volume of glomerular filtrate, that means initial filtrate of a healthy adult is, if you are saying 120, right? 95% of that 120 will be reabsorbed through the nephrons, right? Please calculate and say the answer for this reabsorption. 120, sorry, 95% from the 120. So what is the final volume of the glomerular filtrate after reabsorption? Selective reabsorption. දුව කිත්නුලේ කියද?
එකසිය දාහතරයි.
කියදව?
114යි.
114. ඒ කියන්නේ 114ක් reabsorb කරගන්නවා. ඉතුරු වෙන්නේ just 6 cubic centimeters. නේ? that මේ ටික තමයි ඊළඟ secretion කියන step එකට යන්නේ. But what happens when a person is suffering from diabetes? Right? That reabsorption of the glucose, right, is not completed when a person is suffering from the diabetes, right? So the total glucose amount will not be reabsorbed of a diabetes patient, right? That remaining glucose will be passed out through the urine. Clear? You are clear there?
Clear.
එතකොට diabetes patient කෙනෙක්ගේ urine වල glucose තියෙනවා. හරි? Right. So after that we are coming to the secretion process. Secretion process is also very special. That some materials from the capillaries in the blood of the capillaries, which is there in the nephrons, right, will be secreted into the tubulus of nephrons. ඒ කියන්නේ දැන් කට්ටියක් යද්දී දැන් reabsorption වලින් ආව කට්ටියක් යනවනේ එළියට. ඒ කියන්නේ ෆිල්ටර් රේට් එකට ආව කට්ටියත් ආපහු capillaries වලට ගන්නවනේ. Secretion වලදී capillaries වලින් දැන් ආයෙ එනවා කොතනද? nephron එකට. Right? Hydrogen ions, potassium ions, ammonium ions, creatine, drugs, vitamins. එතකොට urine වල මේ ටික තියෙනවා. Right? Urine වල hydrogen ions තියෙනවා, potassium ions තියෙනවා, හරි? Ammonium ions තියෙනවා, creatine, right? Sorry, creatinine, not creatine. Creatine is a protein, a name of protein. This is creatinine. Creatinine is a nitrogenous excretory product. Then the drugs, honey, vitamin B, right? About vitamin B is a water-soluble vitamin. You know that. You know vitamin B and C is vitamin B and C are water-soluble vitamins, while vitamin A, D, E, K, A නැත්නම් කඩේ K, A, D, E vitamins are fat-soluble vitamins. Now vitamin B is a water-soluble vitamin, so it will be passed through the water with the urine. Okay, do it. Right. So I will ask a practical question. Can urine pass the electricity? Can urine pass?
පුළුවන්.
Why is that?
Salt තියෙන හින්දා.
So ions තියෙන නේද? Right? Free ions තියෙන පුළුවන්. Right? Okay, do it. Clear? Right. Then the removal of urine, urine is released to the pelvis and is transported along the ureters into the bladder. Then the urine is temporarily stored in the bladder and it will be uh, released according to the need of urination. අවශ්‍යතාවය ආවම ඒ කියන්නේ බ්ලැඩර් එක ෆිල් වුණාම යම්කිසි මට්ටමකට වඩා යූරින් පාස් අවුට් කරන්න අවශ්‍ය වෙනවා. එතකොට යූරින් අයින් වෙනවා. Clear? Do so. So you must know the composition of the urine. Correct. Main thing is water and the salt. And the trace elements are creating. Trace means very slight amounts. Right? Right. Uh, right. අපි සහන්සට මැසේජ් එකක් දාමු. පුළුවන් තරම් ඉක්මනට වෙන්න කියලා. Right. හරි. Put the date first and write the topic as process of your urine formation and the first question. Right here. What are the three steps of urine formation? What are the three steps of urine formation? And to the next one. Keep spaces in three lines. Right? Then the second question. What is glomerulus? Third question. What is the difference between afferent arteriole? What is the difference between afferent arteriole and the efferent arteriole? What is the difference between afferent arteriole and the efferent arteriole? Right. Then give just two rules. And to the third question. What is the relationship between the renal artery and the afferent arteriole? What is the relationship between the renal artery and the afferent arteriole? right. ඊළඟට what is glomerular filter rate? What is glomerular filter rate? Next one. Define ultrafiltration. Define ultrafiltration. Fourth one. Fourth one. What is the composition? What is the composition of glomerular filter rate? ඔය හැම ප්‍රශ්නයකටම ඒ දෙන උත්තරේම මතක තියෙන්න ඕනේ දුව. Right? What is the composition of glomerular filter rate? Right? Then to the then we are coming to the selective reabsorption. Right? චුට්ටක් වෙන් කරලා ලියන්න දුව මේකේ තියෙන ප්‍රශ්න ටික. Right. Right. Define selective reabsorption. Define selective reabsorption. Right. Next question. Right. What will be reabsorbed? What will be reabsorbed from the glomerular filter? Next question. What is the balance amount? What is the balanced amount of volume of volume if the glomerular filter rate if the glomerular filter rate if the glomerular filter rate formed during 1 minute of adult is is 120 cubic centimeters. After after selective reabsorption. After selective reabsorption. Right. Next one. What is the difference?
සර් ඒ ප්‍රශ්නේ ආයෙත් කියන්න පුළුවන්.
Right. දුව කොච්චරක් ක්ලියර් එකද? Okay.
what is the balance කියන තැන ඉඳන් if වෙන. එතන තියන්න ටික ඕන.
Alright. If if the glomerular filter rate
if ට කලින් ටික
if the glomerular filter rate දුව එතනට ඒ ටිකද ඊට කලින් ටිකද?
ඊට කලින්
Right. Calculate the balance volume of balanced volume if the glomerular filter rate formed during. Right though?
හරි සර්.
Right. Next one. What is the difference between the urine of healthy adult and a diabetes patient. Healthy adult and a diabetes patient. සේහසදුව මාව ඇහෙනවද? සහසදුව මම ඇහෙනවද? ඇහෙනවනේ. right. දැන් අපි that we have discussed about the urine formation, that the 3 steps of the urine formation, right? And I was giving the questions, right? When you are joining to the session, right? So that I will give you the recording. Then yes, though. You can write the questions from now onwards. Kitnule dua, how many pages should be kept?
පිටු දෙක
Just 2 sides or 2 pages.
sides
Sorry, the 2 sides. Right. That means 1 page, neither. Right. Sehasadua, keep a 1 page and write from this section onward. Our last section was what is the difference between the urine of healthy adult and a diabetes patient? Right?
okay
Right. Then to the next section. So, yes, write it. Define secretion. Define secretion. Define secretion.